And without further ado, I'll start right away because I think I have, prepared quite a number of slides and see how far I can go. Otherwise, I'll cut some of the pieces at the end. The title of the seminar is Pigeon-Pea Breeding, Challenges and Opportunities. And I'll cover aspects related with pigeon-pea production, setting priorities also, and breeding goals. What are the main constraints in pigeon-pea production? Also, how to combine conventional and molecular breeding aspects. Ensure the success of the pigeon-pea hybrids. Aspects of data management, and then just a few words about funding, and finally, what kind of outputs would come out of the program. In terms of pigeon-pea production, the graph here represents the worldwide production, and like you can see, area per se and production have been increasing over time. However, yield has been quite stagnant. The total world area in 2010 was 4.8 million hectares, and in India it was 3.5 million hectares. India produces 70 to 80% of the pigeon-pea. However, it probably consumes near 90% or more. So there is not enough pigeon-pea produced in India, and we import pigeon-pea from places like Myanmar and other areas in Africa. The first thing when you think about the breeding program is setting up the priorities in terms of what would be the target area, target environments, what kind of germplasm base, selection criteria you use, type of cultivars, and also what are the roles and responsibilities of the institute per se, and as well as all the partners, because this is not just ICRISAT alone. In order to set up all of this, we need to take into account the situation analysis and also the desired outcomes that should match quite well with the institutional outcomes. We are now in the process of preparing the pigeon-pea strategy for India. I'm leading that effort, and then the same thing is happening for Africa, so we are defining this in more depth. In terms of breeding goals, the main thing is to increase yield. Increased yield that has to be stable over locations and years, and also maintaining the grain quality. At the end, developing new varieties with higher yield that can be used later on either as varieties or as parental lines of the hybrids. And some of the aspects that in order to prioritize the specific objectives or the selection criteria are related with the agroecological zones that we are targeting, also the maturity groups and phenology of the crop, and also the final use. In terms of agroecological zones in India, for pigeon-pea, there are four major agroecological zones that are listed here in terms of importance. And those agroecological zones are defined by edaphological considerations and climatic considerations. And we also should consider aspects of how is the crop planted. Is it intercrop, sole crop? What are the cropping windows? And also what are the main constraints in each of those agroecological zones in terms of pests, diseases, and abiotic stresses. In terms of maturity, pigeon-pea is a really amazing crop because the range is very wide. If we go by flowering time, we have now varieties that flower in less than 50 days and others that need more than 160 days. And the preference for maturity is also region specific. In some places they prefer short duration, others medium, others long. Flowering pattern, the main two types that are described, even though we talk about some intermediate aspects, the main are the determinate one, where the growth would stop at the flowering time, and then you have the indeterminate one when it continues growing. In most cases, the indeterminate would be preferred because it receives less damage by the insects, but we are still putting effort, even smaller, in determinate types that are desired in some areas that would be more suitable for mechanical harvest and synchronization of maturity. In terms of the use-specific traits, depending on what is the final use of pigeon-pea, and pigeon-pea is really an amazing crop for sustainable agriculture because it has lots of different uses. But considering just a few of them and taking into account the final use would determine what traits we need to target For example, if the final product would be processed dal, we'll need to pay attention to the milling quality and the seed size and seed color. If it is for fresh or canned vegetable, also we'll consider seed size, seed color, pod length. In the case of using the pigeon pea for feed, then protein becomes like an important trait. And if it is for fodder, then returnability and biomass would be traits that we need to consider. The basic breeding approach, and this is kind of simplifying things quite a bit, but so it's straightforward. You identified lines that contain the traits of interest. And this would include even the cultivated lines, varieties, that people really consider the most adapted, the better ones. Also, advanced breeding lines, land races, wild relatives. You make crosses to combine the traits of interest, generate segregating populations using the appropriate breeding methods, and then we apply selection. We can use conventional and marker-assisted selection procedures to reach our target. And also it's very important to do agronomic, quality, disease, pest-resistant trials across locations and across years. In pigeon pea, we are quite lucky. We have many genetic resources, and even right here with us, and there are more than 13,000 cultivated accessions and more than 500 crop wild relatives. As well as for the cytoplasmic nuclear male sterility system, right now there are 7 different cytoplasmic bases for this system. And we can also use mutants that would include even dwarf types, different shapes of the leaves, of the flowers, etc. Within ICRISAT, the germ plasma is being sorted or accordingly, so we can have different levels of depth. And so they have divided into like the active collection, core collection, mini core collection, and base collections. And these collections are being studied in relation to traits, so we have better understanding of what kind of interesting genes could be there. Some examples of the wild relatives that carry useful traits are listed here. There are many more, and in some cases these wild relatives are being crossed with the cultivated ones to develop what is called the crop wild relatives introgression lines, which would be very useful for the breeding program. In genomic resources also in the last, let's say, 5 years, lots of things have happened, and we should be very happy that now we have more tools at our disposal, including more than 25 mapping populations, also tilling population. Molecular markers, quite a number of them, more than 3,000 SSRs, more than 700 SNPs, and more than 1,500 DARTs. Also, there are back libraries, and even the whole genome of pigeon pea variety Asha has been sequenced, so that is available right now. And also there are good transformation capacity available. In this table we have listed just a representative set of some of the mapping populations that we are maintaining within our pigeon pea program, and some of them are quite advanced. We just harvested seed out of the top 3. And the top 2 or 3 will be using, during this year, we'll do a phenotyping for, the second one and the third one. We'll do phenotyping for Fusarium wilt and sterility mosaic disease, and also together with the genotypic information for marker trait association studies. Now, in terms of increasing the genetic diversity at the nuclear level, this is really a big need, because we claim that the base we are working with is quite narrow. So, like I have indicated, there are a number of things. So we have a crop wild relative introgression lines, land races, mutants, elite lines, parental lines, varieties. Lots of things are there. But the breeding program cannot just take all of them directly. Some way of filtering them. So we only use the ones that have the better chances of being useful. We need a way of filtering. One possible way of filtering is with all of them, you do association mapping, for example, that would be really useful to study marker trait association, but also to identify those lines that have more potential for the breeding program. And in terms of the mating and selection schemes that we could use to increase the genetic base of the breeding pool, as well as to enhance recombination, and also for genetic studies, include a number of aspects like biparental crosses that could go directly selfing without selection all the way to recombinant inbred lines, or with selection at each of those stages. So biparental crosses and pedigree based, or we could DEF1 backcross followed by selfing, and then later on proceed with selection or multiple rounds of backcrossing and to introgress the traits that we are interested in. Also, to increase the genetic base of the breeding pool and enhance recombination would allow us to identify good combinations of use for the breeding program, and also to do genetic studies. One of the, this is multi-parent crossing, and this is called the magic population, multi-parent advanced generation intercross, that would involve starting with 8 quite diverse parents and do 2-way cross, 4-way cross, 8-way cross, and then move by selfing all the way to recombinant inbred lines. Another multi-parent type population is what we call the NAM population, nested association mapping. This is again another place where both interest of the molecular genetics and interest of the breeders come together, because this type of population is useful to understand the genetic basis of the traits, but is very useful also to generate lines that contain new combinations that might be of interest to the breeding programs. In this case, Asha, which is the most popular medium duration variety, would be a common parent, and it would be crossed with a number of diverse lines, and after that generate recombinant inbred lines. The diversification at the cytoplasmic level, we have indicated there are already 7 different types of CMS, this is the list of wild species that are involved in this type of CMS. Even we have the A5, the female line is in Cajanus cajan cytoplasm. Out of all of those, we are studying them, but up to now, the one that has shown us the most stable one and the most promising one is the A4 CMS, because the A line has male sterility that is stable across locations and across years. There are also a high number of fertility restorers to make the F1s, and also the fertility restoration of the hybrid is pretty high. One of the aspects in terms of moving along with the breeding program is to understand, out of the traits that are important, what is the genetic basis of the traits, and use that into consideration at the time of developing the breeding schemes and breeding programs. So we need to know how, is this a qualitative or a quantitative trait? How many genes are involved? What is the location of the genes and the position? What is the gene action? What are the interactions at all levels? Intrallelic, interallelic, genotype by environment interaction. Also, what is the heritability of the trait? And what is the percentage of the phenotypic variance that we are explaining with the markers that we identified associated with the traits? In order to estimate the number of effective factors, there are classic methods that can be used, and then there are also like QTL-based approaches that we can use for marker trait association. I'm going to describe some of the important traits for us in the program and a little background of what we know and how can we use some of this for the breeding program. One of the things is flowering pattern. Flowering pattern is quite important. I have indicated that the NDT types seem to be better in most cases because they recover from the damage caused by the pod borers. So NDT types, non-determinate types, are preferred over DT types in most areas. One major gene has been identified as differentiating the DT versus NDT, but there are also some minor genes that modify the expression. And NDT is dominant over DT. So in this particular case, pedigree and backcross methods can be used to obtain the lines that we want, either NDT or DT. And one possible use is that, because we know that NDT is dominant, If we have for the parents of the hybrid one parent DT, one parent NDT, that's one way we can monitor that the parental lines are pure lines. The DT line, all of them should be DT. We should not see any NDT. So it can be used as a morphological marker for testing for homozygosity of the parents. Now, the knowledge of the molecular basis of these traits would also be quite useful, because that way we could identify heterozygotes and confirm homozygotes at early generation stages, and that would facilitate our selection process. Flowering time and photoperiod. The flowering time and maturity are region specific. And for example, in the central zone, south zone is mainly prefer medium maturity, while on the western zone they prefer short maturity, eastern zone it could be short or long duration. So it's region specific. We know that flowering time is highly heritable, and we know that earliness is partially dominant. We also know that in general earlier flowering genotypes tend to be less photoperiod sensitive. And the photoperiod aspect is quite important, because I really believe that photoperiod sensitivity is one of the things that has prevented Pigeonpea from expanded all over the world. And for this photoperiod response, based also on knowledge from other legumes, it's likely that two major genes are involved, so we should be able to target them. Because of the nature of this trait, pedigree-based selection can be used and has been used already. We have to report that lines that flower in between 45 to 50 days have already been obtained and are at very advanced generations and performing very well in places like Punjab and in Almora. They're very happy with those. The knowledge of the genetic basis would allow us to select at earlier stages, and also would allow us to introgress photoperiod insensitivity in medium duration lines. And the maximum yield capacity is within the medium duration, so introducing photoperiod insensitivity there would make a lot of sense. Fusarium wilt, we know that major and minor genes for resistance or tolerance have been identified. Also that resistance is dominant over susceptibility, but also susceptible parents can carry minor tolerant genes. Now, the major genes can be introgressed via pedigree and backcross approaches. And because of this fact that the resistance is dominant, if we want to generate hybrids, with having only one of the parental lines would be enough to develop resistant hybrids. So we could have the Fusarium wilt in the A or B lines, for example, and that would guarantee that you have a resistant hybrid. It would be, in any case, very important to know marker trait associations to better target the genes involved, also know the position effects and interactions. Because we also know that up to 5 races have been reported for this Fusarium wilt, and depending on the races and depending on the genotype, we get different messages. So in order to target things properly, molecular tools would be really important. And also for pyramiding the major and the minor genes, and that way ensuring durability and stability of the resistance. Sterility in mosaic disease, also major and minor genes for resistance have been reported. In this particular case, resistance to SMD has been reported as recessive in some cases and dominant in other cases over susceptibility. And resistance is isolate specific. Major genes can be introgressed via pedigree, base and backcross approaches, but selfing will help to expose the recessive genes, the recessive alleles. Because we say in many cases resistance have been reported as recessive. You will need to self to expose the genes. And it would be better if all the parents of the hybrid are resistant. Also, marker trait association would be quite useful, and in this particular case, SMD already there are at least 2 reports of marker trait association that we can follow. But we know that there are at least 3 different isolates for this virus, and there is different specificity. So we need to take also that into account, that it can be isolated, the resistance can be isolated specific. So in the context of climate change and all of these things, if some of the more aggressive isolates start coming towards us, we need to get ready for that. For example, the Bangalore isolate seems to be quite aggressive. So we need to be sure that we build up the resistance for the, let's say, Hyderabad, Bangalore isolates together and put them together, so also to ensure the durability and stability of the resistance. Pot borers, everybody knows pot borers and pests are the main problem in Pigeonpea as well as in many other legumes. Host plants resistance is actually scarce. There are not many lines that have shown tolerance. There are a number of wild relatives that have been reported as having tolerance or resistance to the pot borers, both Helicoverpa and Maruca, and also crop wild relative introgression lines have been developed, and we are trying to incorporate those in the context of the breeding program. The mechanisms of tolerance or resistance have been determined as being ovipositional non-preference and recovery capacity. We also know, as I indicated before, that the NDT types suffer less damage. So we are trying to focus on that, on the NDT types. Transgenic BT is probably one of the best options to help with this problem. Already there are reports of confirmed effective transgenic resistance. So thinking from a breeding perspective, one of the approaches that would make more sense for us is already use those events that are successful events and backcross those using marker-resisted selection to the parental lines. So that will introgress the BT gene into our parental lines. For then developing pot borer resistant hybrids. The other thing is also we can introgress the BT gene into varieties. And in the case we worry about gene movement, we have a type of flower that is called cleistogamous flower that doesn't open, so in most cases is fully selfed. That would be an ideal thing for putting this BT gene into this type of variety, so gene flow might not be that much of an issue. And at the end, what I really think is BT is likely the best option, but we need to go on the safe side. So combining some of the partial host plant tolerance with the BT would really ensure that there is long-term resistance for the pests. In terms of yield and yield components, we know that both additive and non-additive effects are involved in the yield and yield components, and that the yield components that show the higher levels of heterosis are pods per plant, pod clusters per plant, branches per plant, seed size, seeds per pod, and plant height. So we should proceed with the improvement of the parental lines for yield and yield components, and this will mainly take care of the additive effects, and then development of the CMS hybrids that will benefit from both additive and non-additive. And at the time of developing the CMS hybrids, focus on the performance per se, heterosis, and also on the specific combining ability. Within the abiotic stresses, one that we are working on right now is waterlogging. I think drought might need to be added because terminal drought is also very important in Pigeonpea. We know that the resistance or tolerance to waterlogging is dominant over susceptibility, but likely based on the evaluation of a large array of genotypes, we believe that also minor genes are there because there is a continuous distribution. The major genes can be introgressed via pedigree-based approaches and back-crosses approaches. And because of the fact of being dominant, if we have the waterlogging tolerance in just one of the parental lines, let's say the A for example, that would really allow you to develop resistant hybrids. In any case, also, we are quite interested in studying the marker trait associations to go more in depth into the genetic basis of the trait. And if we have a number of minor genes in addition to these minor genes, also combine, because again, durability is the key, and stability. In this graph, I'm trying to put together a number of things. And in terms of sources of sources that we can use for the breeding program, that include breeding lines, cultivars, land races, crop wild introgressions, mutants, etc. That can be, at times, some of these can even directly be used and select out of the diversity that already exists there for moving into the system of developing new lines, or otherwise used in biparental populations, either pedigree-based populations, backcross populations, or in multiparental populations like the ones MAGIC or NAN, generate large populations and then phenotype them, genotype them as well, if we can, and then proceed with selections. We would generate a number of lines. Now, in order to figure out what is the best approach for hybrid development, identifying parental lines and hybrid development, lines selected, we should involve them in test crosses and in diallele crosses, and take a look at the specific combining ability. Based on the specific combining ability, we can separate lines that combine very well, that could be going into two different groups. The group that would be for the B lines and the group for the R lines, if they have a high specific combining ability. Now, in the case of the B lines, of course, we always need to get the A lines, so that we need to do the backcrossing approach to generate A lines. And then by combining the A line and the R line, we'll get new CMS hybrids that ideally will have the higher level of heterosis, because we took into account specific combining ability to segregate the lines that should go into the restorer group versus the AB type group. I really think in Pigeonpea it's time to move into molecular breeding, and there are a number of advantages. One of them is early selection. The other thing is selection in absence of the disease or in absence of the trait that you need to evaluate, or without doing any test even for fertility restoration. And it's also independent from the environmental conditions, and it allows us to pyramid multiple resistant genes. All of that would reduce time, would reduce the number of lines that we keep in the system, and would also guide selection. Of course, there are always challenges. Some of the challenges involve aspects related with lack of recombination, having traits that are very complex that we cannot really target with the markers, and also maybe even some crossing barriers. But definitely, I think many crops have followed this path. I think Pigeonpea is getting ready to follow that path as well. In any case, phenotyping is really essential and continues to be the most important aspect. No matter how much we do at the molecular level, if the phenotyping is not accurate, we will not get a good message. So we need to combine phenotyping and genotyping very carefully. Now, I have indicated about developing lines that could be used either as varieties or as parental lines of hybrids. I have said that because in some cases, hybrids might work very well, but maybe in some other places it would not work as well. And some of the aspects for consideration are involving like seed production. Not all the places are suitable for production of the hybrids. For the hybrids, we really know that pollinators are required. If that is not there, then you cannot produce seed of the hybrids. Also, can you access fields that are in isolation, more than 500 meters from another field, yes or no? If not, then you cannot produce hybrid seed. Also, technical expertise for hybrids is higher, the cost is medium to high, and the importance of the pure genetic stocks is really essential. And the seed system needs to be formal or well-trained informal. And in any case, for commercial production, in order for the hybrids to be of benefit to the farmers over varieties, the heterosis and for yield has to be at least more than 20%. In any case, for those situations where things can be arranged, hybrids are the great promise. High-yielding, disease-tolerant hybrids, that's one of the biggest achievements in the Pigeonpea program, because there are lots of advantages for the hybrids. In terms of yield, 30 to 40% more yield, more vigor, also greater shoot biomass and root biomass, which would be very good for reducing the seed rate and also for drought tolerance. And the hybrids are ideal for intercropping and also give the farmers more wood, which is another use of Pigeonpea that the farmers really appreciate. I'm going to show you just the top 3 hybrids within the program. This first one, ICPH 2671, has been officially released, and over a period of 4 years, a total of 43 multi-location trials, the yield advantage is near 36% over the medium duration variety Maruti. So that is a very good one, has been already released, and many people are quite happy with it. The other promising one is ICPH 2740, for which variety release is in progress. And over a period of 4 years, a total of 33 multi-location trials, the advantage in yield is close to 38% over Asha. And this one has the best chances likely to move forward because the seed color, everything is very, very well accepted by everybody. And a short duration one, ICPH 2433, also over a period of 3 years, 33 trials. The advantage over the local check for short duration UPAS 120 is near 32%. And here you can see a picture of this hybrid ICPH 2433 out of our station here, ICRISAT. Just checking the amount of flowers, you can probably predict the yield. It's really very good hybrid. Now, in terms of ensuring the success of the CMS hybrid system, there are many things that need to be considered. One of the most important ones is the purity of the parental lines, but also we need a stable male sterility system. Be sure that the heterosis is very good. Also, professional implementation of the CMS hybrid technology, constant training, monitoring, and evaluation. Also, efficient formal and informal seed systems, and marketing and promotion efforts. There is a big challenge for the hybrids. By 2014, KB needs to be sure that the hybrids are in 100,000 hectares. So big challenge there. Now, I said that parental lines and pure parental lines are really the key for the success of the hybrids. What could cause problems or impurities with the parental lines? Outcrossing is one of the main ones. Mixtures, also a possibility. In some cases, non-stability of male sterility system, damage by pests or diseases, also even de novo variation, unintended selection, or even early release of heterogeneous or heterozygotes parental lines could cause that. But again, this is really critical, and we have even went through the effort of developing a checklist that has many different points that need to be ticked to be sure that you reach success with this. And I'm going to only talk about those highlighted ones. Genetic purity, I'll talk about seed production in terms of isolation and the presence of pollinators. Genetic purity. The typical way of testing for genetic purity is visual observation under field conditions. So you take a look at the aspect of the plants, at the morphology, uniformity of height, health, and also at considerations of fertility versus sterility, and draw out the off-types. The other thing which is novel that we are implementing, and this would be very useful for the farmers, is what is called naked-eye polymorphism. There is one mutation of the leaf that is called off-corded leaf. That is when you have recessive alleles, you see the off-corded shape. We have transferred this trait to A and B lines. So now if we have A and B lines, all of them should have this off-corded trait. Otherwise, they are not pure lines. And when you cross with the normal restorer, the hybrid would be having normal leaves. So this is very good. There are some other leaf morphology that we are exploring, because morphology you can tell right away. The other aspect is using molecular markers. Check polymorphism between the A line and B line, and then use the markers that would be diagnostic to confirm hybridity. That is also a very good option that we are already implementing. For seed production, isolation is really critical. Depending on what stage and how much seed you need, you could go just by simple bagging of individual plants, or using nets that don't allow insect pollinators to go in there, or isolation fields with more than 500 meters from another field. Pollinators are really essential, especially for hybrid seed production, and it could be you could pollinate under field conditions naturally, or are there artificial conditions, and they're using cages where you can have hives, or even, sometimes we use our technicians as pollinators. Important considerations in the fields especially are that you have a good insect population, and there is water, trees around, and the weather conditions are also pretty good for the pollinators to feel happy and do the job. We have done a couple of years of experiments of pollination using net cages. There are some good things that we are learning out of this, but also I think the system is not yet perfect. We have Apis mellifera, the Italian honeybee, quite sophisticated. Initially it doesn't like the pigeonpea, it takes some time to get adjusted. We now know that at least they visit the B lines 2 times more often than the A line. Some adjustment still is needed to be sure that they do the right job, or maybe we need to check for some other pollinators. But that is another option if isolation is not acceptable. No matter what, for seed production of the hybrids, some locations have been identified as good locations and bad locations, and in many cases the presence of the pollinators is one of the key factors. So those are things that we can recommend if somebody is interested in producing seed. We can indicate, okay, don't do it here, do it in these other sites. In any case, for the success of the pigeonpea hybrid production, the purity of the parental lines is essential, and now we have tools. Those SSR markers can be used very good for testing for purity, as well as the incorporation of phenotypic markers like this naked eye polymorphism in terms of leaf shape. Could I continue a little more? 5 minutes? Because this is cut a little bit. There is one new area that I really wanted to push hard, and is related with data management, that I think is really essential. And so that requires trained personnel, modern equipment, and also a data management system. This would really allow us to get data and results faster, and also facilitate the decisions in breeding. Trained personnel is really essential to move on this system. Younger people are great. They already know a lot about computers, very comfortable. We really should hire more of them. Modern equipment, we have purchased some of these things. We are in the process of purchasing some others, like single plant threshers, DAL processors, grain counters, data loggers, barcode readers, balances, etc. And this actually became like a very important aspect when we had problems with TFL. We said we need to go more into mechanization, more automatic data collection, because that would really be of great benefit to us. Now, in terms of this data management software or data management system, there were several things that were required. One was that it was a relational database, that we could have phenotypic, genotypic data. It could allow us, from the moment of designing the experiment, crossing blocks, field books, data collection, sharing data, analysis, even for searching, decision tools. It should be friendly and have different levels of control. Some of you have been involved in the process of searching for data management systems, and after evaluating a few, we had decided to purchase AgroBase. So that's the one we are using right now. It took a while, and we are still working on standardization and everything has to be standard and uniform for this system to work properly. It requires time. When it works, then it's quite simplified. So we had to work quite a bit on a number of aspects, including trade dictionaries, etc. Now, having a system like that is really wonderful because you can have inventories of the crossing books, inbred lines, segregating populations, everything that you want stored there, multi-location, multi-data, easy accessible by all of your colleagues. Immediate benefits. You can save time, money, improve efficiency, and reduce errors. For example, for data capture, just collect directly and then transfer directly to the computer. And you don't need assistance for going from paper to computer. Some curation and cleaning of the data is really required, but there are also some validation tools that we can use. Templates, once they are ready, you can reuse them, you can save paper and ink. And additional benefits is that the data is immediately available for analysis. The data analysis is simplified because it's connected with a data analysis software like GenStat, and also it allows you to make faster decisions. Okay, we'll cross this one with that one because we already know it's good. Some of the outputs of this data management includes new discoveries that can be based on exploring old data or historical data. Information for use in new data-driven research, preserving the scholarly outputs, facilitate data sharing, but also facilitate queries. If I want to know how many lines have resistance to SMD, Fusarium wilt, and Phytophthora, I could just do a query and come up with that. And also more and more, this is the requirement of some of the funding agencies, because they need data management, also retention of the data and access to the data. This would be what we are doing with AgroBase is linked or related to efforts by the Generation Challenge program in terms of this integrated breeding workbench that goes from the moment of planning the experiment all the way through the final decision making. And now with more of the molecular tools that would be also integrated into the system, so all of the pieces will be there, phenotypic information, genotypic information, data analysis, to really make faster decisions on this. Okay, in any case, I'm going to kind of wrap up with some of the outputs that could come out of the Pigeonpea program. And this is not only ICRISAT, of course, we have the partners at the national system, international, NGOs, etc. But some of the things that are coming out of the program and should continue coming out of the program include improved pure lines that can be released directly as varieties, improved parental lines. Also for the case of the hybrids, because we want to know more about the heterotic groups and about fertility restoration, we should be able to identify the best hybrid combinations much faster and efficiently. Also genetic knowledge of fertility restoration, heterosis, Fusarium wilt, SMD, and waterlogging. The CMS hybrid production technology, it's already well established but needs to be shared with the partners. Molecular breeding needs to be implemented in the system. We have initiated some aspects already. Data management, I think, is essential and we'll continue with that. We're also well placed for doing formal courses in breeding, genetics, and seed production, as well as for training technicians, farmers, graduate students, and scientists. And one of the aspects that is coming more and more now is the consultancy demand. In terms of funding, I'm not going to say too much, but just a couple of things. Is that being Pigeonpea a crop that is not worldwide distributed, I have found myself sometimes difficulties of getting international funds because they say Pigeonpea is not a crop of importance to our country. The way of going around that to be able to get international sources of funds is to focus on climate change, things like drought, waterlogging, and also in terms of poverty alleviation, whatever is linked to technology transfer. In that case, I think international funding is possible. And we have lots of support from the Indian government for that. We can continue on that regard in terms of we can push on that the molecular genetics, molecular breeding, and also the expansion of the hybrids. And thank you very much. I think with that I'll end. Just in case.
Things you mentioned we have been discussing in the past. One was the integration of BT, of course, into the AV line. So we need to make some sort of a decision on how exactly to pursue that. And we could start the optimization work. We've been discussing also with Dr. Saxena on that. And second, I think I'm more curious about, we discussed when we joined earlier, was using some sort of a marker system like either Christogamous flowers or something else, maybe flower shape for inducing, for transgenics basically, so that they can be easily identified in the field conditions. So I think these two, we can pursue this further. But I'd like to hear more, what are the opportunities available, especially using Christogamy or any other morphological trait to associate with transgenics?
I definitely think that having some type of morphological marker would be very useful to confirm that you have the transgenic. It would help certification agencies as well. The case of the Clistogamous flower, I think it could act as both, but that is only for varieties. It could act as a marker to identify that you really have the transgene, but it could also act as a way of preventing gene flow, because it would be almost close to 99%, 100% selfing. So for varieties, if varieties is one way, Clistogamous is definitely the way I think to go. And the leaf shape of this of corded, for example, we have already A lines and B lines, and we have not committed them yet to hybrids. So if we are sure that BT Pigeonpea is there, we can reserve that type of trait for that specific use and try not to use it in other contexts.
So that's a decision you should be able at some level, some stage. Next.
Hilde, what is the scope to improve the harvest index of Pigeonpea, because it's very low and probably one of the target to.
I think you are totally right. You are on the dot there. Increasing harvest index would be really what you really need for increasing yield as well. So we should definitely take a look at that.
Isabel, you made a mention that B lines would be identified with high specific combining ability. Is that right?
I have indicated that based on the diallele crosses and on the line by tester crosses, if you check the specific combining ability, based on the specific combining ability, you could divide the lines involving the hybrid into two groups based on the specific combining ability.
No,
so it could be into the R group versus the A/B group.
Because the specific combining ability by definition is for the combination,
yes,
and not for the lines. Not for a particular line, because if you see, by definition, it's the deviation from expected combining ability,
yeah,
for a combination. So in that case, because the specific combining ability would be for hybrid combination, not for a line.
yeah.
For lines, it's general combining ability.
Right. No, it's for lines, but I'm talking in the context of the hybrid. Once you make the hybrid, you know which two lines that are involved in the hybrid that give you the highest heterosis. So then you can, based on that, the division of the lines, start thinking about heterotic groups.
And give opportunity for socioeconomics, Partha and Ashok, and then secondary.
You have shown that the hybrids are 30% higher yield. I thought it's also important to look at its acceptance along the value chain, in the sense of traders, wholesalers, processors, and ultimately the consumer.
Mm-hmm.
Price is one of the indicators which will tell you whether it's acceptable along the value chain. So in that context, I just wanted to mention that the traits that you mentioned, you have not really mentioned market preferred traits, which I thought was missing. For example, in Pigeonpea, cooking time and taste, and some of these, the pericarp on top, and the hull, how thick it is. Some of these things are traits which the market looks for. And I think in the breeding program, these should be built in and looked at.
Definitely consumer-driven traits are really essential. This is one of the things that also came in my previous job, that initially we were only based on or using traits that are of preference for the farmers. But later on, I think these things come once the projects mature a little more. You start focusing also on what are the consumer preferred traits. At this point, because of the cost involved on those evaluations, typically we only do those evaluations with materials that are quite advanced. But likely we should start earlier on, so we would not keep in the middle things that are not that acceptable. Up to now, I don't think we have encountered situations where some new lines or hybrids are rejected because of the fact that they don't have those traits preferred by the consumers. But definitely we should take a look at that. Actually, I have to just say, today I went to see how our new dial machine is working. So we'll be looking into dial recovery and all of those things. We have also distributed bags of dial with questionnaires for getting feedback. But if we see the need and there is consensus that it is very important, then we have to more systematically be sure that we first listen what the consumers need to say in terms of traits that should be considered, and then actually perform those tests to have the information.
Sure.
Isabel, you mentioned that photoperiod sensitivity influences the spread of Pigeonpea. How it influences, that is one thing. Number two is what specific measures you or your group is taking in addressing the seed production issues in the current CMS hybrids.
What?
Seed production issues. Hybrid seed production
issues. In terms of photoperiod sensitivity, the thing is that if you take medium duration or long duration hybrids to higher latitudes, let's say even to Europe, 42 degrees north or whatever, they would not flower or mature, or it would take forever to flower or mature. So obviously they would not fit the cropping systems there. On the other hand, if you had materials that had are less photoperiod sensitive, they could be planted at higher latitudes and could be expanding. That is just my guess, because in Europe or in the US, you don't talk about Pigeonpea, and I was wondering why. And then if the thing comes, okay, even though if you bring it, it would never flower, it would never mature. So that is just one of the possibilities there. In terms of seed production issues, what measures we are taking to ensure that that is properly done? We have a seed production expert that could probably further elaborate on that. But part of that is the training, get involved with the farmers and with the private sector so they really know how the CMS system works and what are the precautions that need to be taken. And also we interact with the farmers and with the seed sector in terms of monitoring, evaluating, and being sure that is happening properly. For the varieties, what has been working quite well is the one village, one variety concept. We are wondering maybe something like that, similar, could be implemented for the production of hybrids. Any other thing, Myers, you want to add?
I think we should not trouble Meyer.
Yeah, he's KB.
Thank you. I think very important questions. I think what we are trying to do at the first stage is to identify where we can grow parental lines properly and hybrids properly. This is the key factor we have learned last 4 or 5 years. So we have been troubling ourselves a lot. Now we know where to grow. See, for example, we have identified many locations in Madhya Pradesh where Pigeonpea is not grown at all. It's not a crop there. But once we grow hybrid production, yields are more than 1 ton per hectare. Unbelievable yield we are getting repeatedly there. So that's one thing we are doing. Second thing we are doing, we are putting more emphasis on purification of breeder seed here. And we want to track initially, for example, next 5 years, the challenge at Isabel is very high with us. to grow Pigeonpea in 100,000 hectares and with quality. So we have to do really a good job on that, and we have trained a lot of people in our collaborator universities and all that to produce high quality parental seed first, and then follow up these other qualities and go for large scale testing. But training is very, very important. Next week also we are getting some trained farmers here for training purpose. So we are doing all the stages, our training activity, seed activity, then also we are trying to use the molecular marker now. So we have already talked to Rajiv, and then he's working out cost, how much cost it will involve. Basically we'll do primary seed characteristics through that, and then definitely other things will be followed as in other crops. Thank you.
Dr. Hari Sharma and then Dr. Legumes. After that, the DG will have the last word. So
Okay, Isabel, I'll desist asking a question about Bodborus, but you listed a number of these CMS sources. Is that classification based on differential restore response, or it's based on just source of the CMS?
I think initially source of CMS, source of cytoplasm, I would say, because then in terms of restorers, there are some restorers that could restore across CMS systems, but others only restore for one system but not for the other.
Dr. Legumes.
Thank you, Isabel, for a very exhaustive seminar and learned quite a bit about Pigeonpea. It's not a question, it's a general comment. And as you pointed out, that the yield is stagnant in Pigeonpea, and last 35 to 36 or 38 years, right at ICRISAT, we have been working on this crop, besides Indian national programs and others. In spite of that fact, the yield remains stagnant. Only two major achievements I can think of in this particular crop. One was short duration Pigeonpea and CMS hybrids. These are the two significant achievements in Pigeonpea past 60, 70 years. What I was expecting in this seminar, a critical review of the past program, where did we go wrong in the past and how we could rectify that mistake. Particularly, the conventional component of your seminar, convergent crossing, magic population, these are the new words for the old wine. So there's nothing new to me in that one. So I still don't get an idea how you are going to succeed if you continue to follow what was already done and abundant without much progress.
I think post-plant aspects is just one of the things that you need to be sure that the yields go up. There are many different considerations and aspects. One of them is even access to water, access to fertilizer, access to herbicides, pesticides, and also agronomic practices. Despite the fact that we recommend spacing plant to plant, row to row, people still do broadcast. Some of the conditions where the Pigeonpea crop are grown are really difficult, marginal lands. I mean, we need to really address many other components to be sure that once you put everything together, you get a good result. Because the yield potential of varieties that have been developed and hybrids, the yield potential is very high. But there is a big gap between the actual yields and the yield potential. By improving the yield per se, you can help a little bit pushing that up. But in order to make a significant difference, there are many other aspects that should be really taken into account. And that's the concept of the systems, systems work, where many scientists from many disciplines work together to put a package that would ensure that the yields go up. But in terms of this, what can we do on our hand, increasing the genetic diversity, the genetic base of the breeding programs is really essential, because if we don't have that, then we cannot go up. So that is number one thing.